should probably share my screen first, huh? That might help. How was your morning started?
It's been pretty good.
Pretty good? Good. Pretty cool. I received a text this morning from Ruth Hodabella. She's sitting in her chemistry class, and they're talking about spreadsheets. And she's like, Manro, I can't believe that people don't know how to do spreadsheets. You and I will talk about spreadsheets. If you don't know how to use Excel or Google Sheets, spreadsheets, we will talk about those in about 2 weeks as we start digging into your IA and you start handling and processing data. That's coming. Super cool. All right, so when we are infected with a pathogen, that pathogen is then eaten by our immune system. That's how your immune system protects you from infection. You literally have cells called big eaters, macrophages, who come in and eat the infection. Well, to make the macrophages more efficient at eating the infection and having the infection be knocked down much sooner, there are these Y-shaped proteins called antibodies. Antibodies are not alive. They're Y-shaped proteins whose shape is very, very specific. The receptors on antibodies are specific to the surface membrane proteins on the pathogen. Okay, so be sure that you grasp that the receptors on antibodies match the shape of the surface membrane proteins on the pathogen. So the antibodies then bond to the pathogen, but the antibodies aren't what destroy the infection. All the antibodies do is they mark the pathogen chemically, and that attracts the macrophages to then eat the infection. Okay, so when people say antibodies protect you, kind of, okay? If you don't have the macrophage, all right, the antibodies would bond to the pathogen, and that's all that happens, okay? So you have to have the macrophage come in and eat the infection, and the antibodies make that phagocytic process much, much more efficient. So see this as the big picture, okay? There's that saying, sometimes you can't see the forest for the trees. Sometimes all these details and students get overwhelmed with the details, so back off, all right? And there's 4 big steps. It's macrophage, that's 1, helper T cell, 2, B cell, 3, and then differentiation, 4. There's 4 major steps here. The macrophage eats the pathogen, presents the surface membrane protein, 1. The helper T cell then carries that surface membrane protein, that's 2. The helper T cell then sensitizes a B cell, 3. That B cell then differentiates into long-term and short-term cells, okay? So are you comfortable with primary response?
Yes.
All right, okay, so then this is only the first time you see an infection. This is when we get sick. This process takes, depending on your age and your metabolism, 5, 7, 10 days, okay? So while you're sitting there feeling terrible, you're waiting for this to happen, all right? And then once the antibodies start being produced by the plasma cells, the macrophage starts to eat the infection, and your viral load or your viral concentration starts to drop. That's why I feel better today. I feel better. I feel better. That's the antibodies bonding to the pathogen and your immune system eating the infection. All right. Then we have secondary response. Have you ever had the sensation where people say, I feel like I'm trying to get sick, where you'll get that dull headache behind your eyes, right? Your skin will feel just a little achy. Maybe your throat feels kind of dry or kind of rough or a little sore, and you go, I'm getting sick. And then you'll go to bed and you'll get up the next day and it's gone. Have you had this experience?
Yeah.
Okay.
Yeah, yes.
What that means is that was a secondary response. It means that there was some pathogen, some bacteria, some virus that invaded your body. Your immune system recognized it, responded quickly, usually within about 24 to 36 hours, and then your immune system was able to eat the infection before the viral load or the bacterial concentration got high enough that you got sick, okay? So this is secondary response. This is when you are exposed to a pathogen the second time, something that you have already seen. So earlier here on the primary response, We tend to focus here on the short-term cells, and we don't really focus on the long-term cells. We just say, and they make memory cells, okay? Secondary response is totally based on these memory cells, okay? So what these three are is these are all memory cells. However, these memory cells have different, it's called specificity. And you say, specificity, what are you talking about? If you look really close at the shape of that receptor there, and the shape of that receptor there, and the shape of that receptor there, I wish Campbell's would have done a little bit better job here of making these receptors more obvious. Okay, whoops, didn't mean to do that. There we go. Oh, there we go. So can you see, it really doesn't come out even here. This is in the shape of a W, and this is in the shape of a U or a cup, and then this is more square and it's deeper. So what Campbell is trying to show you here is that this receptor is different from this receptor, which is different from this receptor. So these are memory cells that are sensitive to different surface membrane proteins, okay? So what you have here is you have an antigen. What you have here is surface membrane protein. So some pathogen or some antigen that your immune system has seen before, and so your immune system has produced memory cells in response to that previous infection. You are then exposed to that pathogen again. The macrophage presents the surface membrane protein, and the memory cell then that's specific, that says, chemically, I recognize that, okay? And what this does is it causes activation. Activation of a specific memory cell. Okay? And this activation leads to mitosis. Okay, so you have some memory cells, but when they get stimulated, they reproduce in very, very large amounts. And these memory cells, don't forget, are a type of B cell. Well, do you remember what B cells have the ability to do? What did the B cells have the ability to do?
Differentiate.
Differentiate, okay? These are a specific type of B cell. So these B cells then, once they respond to this infection that you've seen before, they differentiate. What do you think they differentiate into? What's over here?
B cells that recognize the
B
antigen.
cells that, yep, B cells that recognize the infection, which we call
Memory
memory
cells.
cells. Yep. Which again are long-term immunity. This is where your immunity comes from. What do you think these are?
Antibodies.
Yes, I agree. These are antibodies, and antibodies are made of what kind of cell?
Plasma.
Made by what kind of cell?
Plasmid.
Plasma cells. I agree. Which are short-term. And so then these are the antibodies here and the antibodies here. So you see an infection for the first time. When you see the infection for the first time, your immune system produces plasma cells, so it can produce antibodies, so it can knock down the infection within 5, 7 to 10 days. All right. The first time that you see a pathogen, you also make memory cells. Why? So then you have biochemical and cellular memory. This is cellular immunity. Okay. So therefore, this memory cell waits. And if you are exposed to that same pathogen again, that activates that memory cell then goes through mitosis. It replicates into even more memory cells. That's long-term. Or short term because you've been exposed, and this is why you only have that little sore throat for about a day, or you get that dull headache, or your skin kind of feels weird, because this process here only takes about 24 to 36 hours. You then flood your lymphatic system with antibodies that triggers the function of the macrophages, and you eat the infection before it reaches a high enough concentration for you to actually get sick. Okay, so do you think it's a fair essay question to ask you to compare and contrast primary and secondary response?
Yes.
Yep.
Absolutely. IB loves to ask that question. Okay, they will ask you, what are the similarities and differences, compare and contrast, primary and secondary response. Okay, so chickenpox. How is your generation different from my generation with chickenpox?
Your generation actually got it, and we just got vaccines.
You got a vaccine, you bet. Okay, when I was a kid, if somebody in the neighborhood got chickenpox over summer vacation, do you know what the adults would intentionally do?
They would bring other kids so they would get it.
Exactly right. Okay. If it was during the regular school year, they tend not to do it because, you know, chickenpox will make you sick for about 7 to 10 days. But if it was summertime and we had the, you know, that big chunk of time off, seriously, they would intentionally get the children together so they'd all get chickenpox because it was summer vacation. All right. So then we would be infected by the virus and we get a fever and our skin would outbreak. And I remember having chickenpox, right? And I made antibodies the old fashioned way, right? I had the infection. Well, how do vaccines work? Vaccines inject you with either the inactivated form of the pathogen, or they simply inject you with the surface membrane protein. Your immune system then recognizes that and produces antibodies to that infection. So the majority of you formed memory cells based on the exposure you got from the vaccine. That's how vaccines work. The function of vaccines is actually extremely simple. It sensitizes your immune system so that you produce antibodies to a pathogen that you've never had to survive an infection from. Okay? Measles, mumps, rubella, diphtheria, whooping cough, pertussis, chickenpox, which is varicella. These are all infections that used to kill thousands and thousands of children. Okay, up to about 1920, right? We're only talking 100 years ago. Up to about 1920, the mortality rate of children in North America was 30%. A third of all children died before reaching the age of 8. Okay, and then they came up with two huge things: antibiotics and vaccines. All right, and now for a child to die of a childhood infection is actually extremely rare. Okay, and we'll talk more about vaccines, and we need to talk about how people are critical of vaccines, specifically the MMR vaccine, the measles, mumps, and rubella. We will talk about a gentleman named Whitaker who published a study that had a statistical error in it. There's a lot to talk about. Okay, so comfortable with, yes. For the most part, that's true, okay? Unless there's a mutation in the virus and your immune system doesn't recognize it, okay? So there are some viruses that mutate at an extremely high rate, okay? So something like influenza. Influenza mutates just crazy fast, okay? And it mutates so fast that the vaccine that we made last year doesn't sensitize you to the current version of the flu. So what has to come out every single year?
A new vaccine.
A new flu vaccine, okay? But in 1954, Jonas Salk came out with the polio vaccine, and we still use the same vaccine today, 60 years later, almost 70 years later. What does that tell you about the mutation rate of polio?
Slow.
Super slow. All right. So vaccines protect you from the most common form of the virus, but not every form of the virus. So what Ms. De La Cruz just asked was about chickenpox. Once you've had chickenpox, will you get it again? For almost everyone, no. But if you encounter a form of, it's called varicella. If you come in contact with the varicella virus, it's genetically different enough, there's enough of a mutation that your immune system doesn't recognize it, you can get sick with it twice. Just because you've been vaccinated or you've been sick with it once doesn't guarantee that you won't get sick with it again if it's genetically different enough and your immune system doesn't recognize it. It's a good question. Okay.
Mr. Mario,
Yes.
like a lot of people, they don't get like the flu shot because they say that you're gonna get sick afterwards, or like you get sick even if you have the flu shot, and like you come in contact with someone who has the flu, like you still get sick. Why does that happen? Or like why? Yeah, why does it happen?
Yep. All right. So if you get the flu vaccine and you still get sick with the flu, that's a definite possibility. What that means is that you were exposed to and infected by a form of the virus that is genetically different from the form of the virus that the vaccine protected you from. It doesn't mean the vaccine doesn't work. The vaccine works against a certain number of the viruses. No vaccine 100% because there's genetic variation in the population. There's genetic variation in populations of viruses too. So let's see. Some of the females. You may have been vaccinated with the oh my gosh, Gardasil. It protects you. It vaccinates you for HPV, human papillomavirus. Do you know of this vaccine?
Yes.
Yes.
Okay, so the Gardasil vaccine protects you from the 3 most common forms of HPV. There's 6 different forms of the virus. Okay, so if you read the literature, it says that no vaccine is 100%. It's about 90 to 95%, but no vaccine is 100% because you don't know about all of the genetic variation in all of the viruses of that family on the surface of the earth. That's not possible. So when someone says, oh, I got a flu vaccine last year and I still got sick with the flu, so the flu vaccine is garbage, doesn't work. I go, no, I understand your science, okay? The vaccine works against the form of the flu that is most common. You encountered what we call a variant.
Okay,
Yeah.
that makes sense.
Yep. And so what's going to happen is as with any other science, as you get into the science and you hear people say things about science that are inaccurate, you have to make a decision. You know, do I tell them what they're saying is wrong, or is it better for the relationships to I'm just going to keep my mouth shut? Yeah, and regardless of your area of expertise, you will run into that where people say things that are incorrect in your area of expertise, and you've got a decision to make. Yep. That's also why there's discussions about we should fast track the COVID vaccine. If you hurry it, that limits the variation of the COVID that we can protect you against. So if you hurry the vaccine, you have limited protection. That's the bad news. What's the good news? You get a vaccine early. Okay. All right, let's wait. Usually vaccines are developed over 2, 3, and 4 years. So if we wait 2 and 3 and we all lock down for 2 and 3 and 4 years, the vaccine that comes out will be about 95, 98% effective. Yay. But what's the bad news? You had to wait 2 and 3 and 4 years. So that's the decision that bio companies are making right now. That decision is being made by pharmaceutical companies today. What do you do? It's a really, really important decision.
Mr. Manro,
Mm-hmm.
where did the idea that vaccines can cause autism come from?
Came from a gentleman named Whitaker. We will hit that. Do me a favor, write that question down. I promise I will answer it when we get into vaccination. Let me think where we talked about vaccination. We talk about vaccination really soon. Where do we hit? We are going to talk about a gentleman named Jenner that came up with vaccines about 150 years ago. And then there's another gentleman named Whitaker out of England who did a study on 12 children and published a study that was mathematically incorrect. and the media grabbed a hold of it and published it. And since then, Whitaker has retracted his publication. We have pointed out the mathematical error in his statistics. We have pointed out that his study was only on, get a load of this, 12 children. That sample size is crazy small, and yet the general population still holds on to the fact of what he published in the 1980s. And it has been shown repeatedly to be a mathematical error and has never been reproduced again. And people still hold on to it. And I will get you all of those details when we talk about vaccines. I promise. It's really important, especially now. Yep, that's where the whole anti-vaxxer movement comes from, and I have to address that because it's not one of those issues where, oh, you know, it's a political issue depending on how you see it. No, Whitaker's science is incorrect. Whitaker's statistical analysis is incorrect. You say, well, then why is there a movement? Well, why do people think the Earth is flat, right? There was a flat earther last year that got enough money to shoot his butt up into space because he was going to take pictures and show that the Earth was flat. And so they shot him up there and he pulled his camera out. And guess what he took pictures of?
Around Earth.
Around Earth. I love that. That was fantastic. He came back down and he went, the Earth is round. And we're like, yeah, that's awesome. Okay, so that should take care of 3C and 3D. Yes, and yes, and yes. All right, so let's talk about 3E. 3E says viruses lack a metabolism and cannot therefore be treated with antibiotics. So let's hit 3E. I'm going to slide that up just a little bit. It's too close to me. It's uncomfortable. Okay, so antibiotic. Anti. What does it mean to be anti?
Be against.
Again, sure. And then bio is the root for life. So when you look at the term antibiotic, what does antibiotic literally mean?
Against life.
Against life. However, specifically, what kind of life? Against prokaryotic life. And of course, when we're talking about prokaryotes, we're talking about bacteria. Okay. So there are a couple of common bacteria. Erythromycin, I need to check on the spelling on that. It's E-R-Y-T-H-R-O, E-R-Y-T-H-R-O-M-Y-C-I-N, erythromycin. This is an extremely common antibiotic. And what antibiotics do is they kill bacteria. But how do antibiotics kill bacteria? Antibiotics kill bacteria by disrupting the biochemistry of the bacteria. Okay, so erythromycin, what it does is it bonds to the ribosome and blocks protein synthesis. Well, if the bacteria can't make proteins, what happens to the bacteria?
It will die.
It'll die. Okay. Grove is knocking on my door. Okay. He's texted me like 3 times in the last minute. I'm like, dude, chill out. All right. So the antibiotic disrupts the biochemistry of the bacteria. All right. But do viruses have a biochemistry? Do they have organelles? No. So are antibiotics effective against viruses? No.
No.
Okay. All we can do with viruses is treat the symptoms. So penicillin. Penicillin comes out in the 1940s. Extremely common antibiotic. We've been prescribing it. I live with crazy people. Do you know that?
Hi.
I live with crazy people. Watch crazy people. Go over.
Hi.
We're talking about penicillin.
Oh.
You missed period 1 where we're talking about they came to the realization that flies eat poop, and therefore flies are detritivores.
I love freshmen.
Freshmen are awesome.
They're great.
We had some extra time, and so I was asking them if they had any questions on their exam or their lab write-up, and somebody asked about decomposers, and then they started talking about detritivores, and in about 30 seconds, we were talking about how flies eat poop. It's awesome. All right. So what penicillin does is it disrupts cell wall synthesis. And so what happens is bacteria have a cell wall, not all of them, but some of them. And so when you expose bacteria to penicillin, the cell wall is incomplete. And what happens is in bacteria, they have an extremely high cytoplasmic pressure. And so what happens is the plasma membrane then herniates out or is driven out of the cell wall like such, and the pressure gets so high, what do you think happens to the plasma membrane? Because the pressure is so high.
It ruptures.
It ruptures. And then the cytoplasm flows out, and what happens to the cell? Cell dies. Yes, it does. It dies. I agree. Okay, so this is how penicillin works. So referring back to natural selection, these antibiotics, this has been around since the 1960s. This has been around since the 1940s. All right, so penicillin has been used for now 60, almost 80 years. Is this a pressure of natural selection?
Yes.
Yes. What happens if you apply the same pressure of natural selection to a population over and over and over again? What eventually happens?
Mutations lead to immunity.
There you go. Okay. The genetic variation in the population, like mutations, allow the individuals that are resistant to then survive and reproduce, and your population becomes resistant or immune over time. That is basic evolutionary theory. So when people say, "Oh my gosh, antibiotics don't work because the bacteria they're adapting," we go, we call that evolution. Okay, evolution even applies to bacteria. Evolution applies to all living things. Okay, so big picture idea: how do antibiotics work? What do they do? What do they disrupt in the bacterial cells? Yes, the protein synthesis, the biochemistry of prokaryotes. So when you take antibiotics, do they harm your cells?
No.
Nope. Now, if you take antibiotics in high concentrations, they can cause diarrhea as you disrupt it's called your intestinal flora. You have populations of bacteria in your intestines, and when you take large amounts of antibiotics, that kills the bacteria in your intestines and can give you some really bad diarrhea. So if you ever have to take antibiotics, you may want to eat more yogurt or other what they call probiotics so that you can repopulate your intestines. All right, so that's bacteria and antibiotics. Now we need to talk about viruses, because are antibiotics effective against viruses? No. Antibiotics have no impact on viruses at all. Okay, this is why usually it's good news that we live this close to the border. Sometimes it's bad news, because people will get sick and they'll go, "Oh, I don't want to go to the doctor in the States because then I have to pay for the doctor's visit, and then they have to give me prescriptions. I can go across the border and I can buy the antibiotics over the counter." And so they take the antibiotics and then they don't work if it's what kind of infection?
Viral.
If it's a viral infection, okay? You can't tell the difference. You can't sit there and go, "Oh, today I can feel it. I'm sick with a bacteria." Or, oh, today I'm sick with a virus. You can't tell the difference, okay? Unless the bacteria attack you or start to grow in your tonsils, and you get these nice packets of bacteria. Then you go, hey, look in the back of your throat, okay? But if you get an infection in your sinuses or you get an infection in your chest, you can't sit there and go, yeah, this one feels viral. Okay, you can't tell. All right, so let's talk about viruses and why antibiotics are ineffective against viruses. So a virus, let's talk about the structure of a virus to start with. Viruses are so efficient. They're beautiful in their simplicity. All viruses have a protein coat on the outside, all of them, okay? And then coming off of that coat, there are receptors, like such. You know how we talked about the surface membrane proteins?
Yes.
Yes, these are the surface membrane proteins. These receptors are surface membrane proteins. I should stick a little receptor in there just for balance. There you go. Okay. And then inside of the protein coat, there's genetic material, like such. You guys can make noise. It's okay.
Okay.
I'm presenting from our home. If you guys are in our kitchen and making noise, that's okay. They're in the kitchen right now, like trying to be really quiet. It's okay. Make some noise. Yes. Okay, so this is genetic material. Now, what's interesting about viruses is this genetic material could be DNA or it could be RNA. There are two different classes of viruses. There are DNA viruses and RNA viruses. So what you see here, this is a virus. And in this example, this is a DNA virus. We will talk about RNA viruses in in the 19.8. Okay, so you have a virus. Now, there is going to be some receptor here. And this receptor matches the receptor on the virus, okay? So if this virus had a receptor shaped like this, then the cell would have a receptor that matches that shape, okay? That's why viruses are tissue specific. Hepatitis only affects or infects the liver. Influenza only infects the lungs. HIV only infects the immune system. Why? Because the receptor on the virus matches the receptor on that cell type. That's a really important factor about viruses. Viruses have these surface membrane proteins, they're receptors, and the receptors on the virus match the receptor on the host cell. That's why viruses infect specific tissues. You'll never hear someone, and they're coughing, and they go, "Oh, are you sick?" And you go, "Yeah, I got hepatitis." Hepatitis doesn't infect your lungs. Why? Because the receptor on hepatitis matches the receptor on your liver cells. That's why viruses are tissue specific. Okay? All right, so what happens here? You get entrance into the cell. What literally happens is, since the virus bonds to the cell literally invites the virus in. The cell doesn't realize that this is going to end up killing the cell. The virus bonds to the cell then opens its plasma membrane, and the virus is chemically invited in, okay? And since this was a DNA virus, what is injected into the cell is viral DNA. And then two things happen. You get replication and transcription. You get transcription and replication. Well, if you take viral DNA and you replicate viral DNA, that makes more copies of what?
Virus.
Viral DNA.
So what viruses really are is they're molecular pirates, okay? And they come in and they take over the biochemistry of the host cell. The host has all of the enzymes necessary to reproduce the virus. The virus doesn't have its own organelles and enzymes to make this process happen, okay? So viruses take over the biochemistry of the cell. So the virus is chemically invited into the cell. It injects its DNA into the cell, takes over the replication process of the cell to make more viral DNA. It then injects its DNA. Transcription then produces messenger RNA, but what kind of messenger RNA?
Viral.
Viral messenger RNA. Then that viral messenger RNA is read by the process of translation. Again, the cell's own ribosomes are taken over to make viral proteins. And then this viral DNA and viral proteins are assembled in what we call, really fancy name, virus assembly or viral assembly. And then what is produced?
More viruses.
More virus, okay? A virus's whole function is to take over a host cell and turn that host cell into nothing more than a virus factory, okay? And we call this process of the host cell producing more virus, we call this viral shedding. And the cell will do this, just shed virus and shed virus and shed virus until, since its own biochemistry is taken over, what eventually happens to this cell?
It dies.
The cell dies, okay? But not before the cell has replicated millions of viruses. And then the viruses, or the virus, then lands on the tissue next to it, invades that virus, and more viral shedding, and then more and more and more and more and more. And this is why you get more and more sick. You know, this is why, oh, I don't feel good. Oh, I don't feel, wow, I really feel bad. Why does the infection make you feel worse? As more and more of your cells are killed, okay? And then you feel super bad. And, oh, I just feel so poor. And then all of a sudden, one day you wake up and you go, hold it. I feel a little bit better today. Why do you turn that corner biochemically? What happens that makes you feel better?
The macrophages start eating a bunch of the viruses.
Yes, primary response, and the good guys show up and they start eating the bad guys. This is literally good guys, bad guys, okay? It's a gang fight is what this is. It's our gang against the other gang, okay? If the other gang wins, we stay sick and it could be life-threatening. If the good guys win, if our gang, then you recover and you go, whoo, last week, boy, I was really sick. And it's all biochemistry.
Mr. Banner,
Yes.
once the virus takes over the cell, can the macrophages do anything to it?
Yes, there is a whole host of white blood cells, and they're called cytotoxic T cells. I should write that out for you. There's another form of white blood cell that we don't talk about in IB, but you will talk about in college. They're called cytotoxic T cells. What their job is to identify the cells that are shedding virus and kill them. These are your own white blood cells whose job it is to kill your sick cells. These cells also kill cancer cells. These are good guys. These are the police, right? That kill the bad cells. Okay. However, if these cells go out of control and these cells kill healthy cells, you ever heard of lupus? That's what lupus is. Lupus is an overactive immune system. Lupus is when your own cytotoxic T cells, rather than killing infected cells, they kill healthy cells. And depending on which healthy cells these cytotoxic T cells kill, changes your symptoms. Okay, these cells might attack your skin, and so your expression of your lupus is maybe just a really bad skin rash. If these cells turn against your nervous system, they can destroy your brain and kill you very quickly. So this is, again, as everything in biology is, it's homeostasis, it's balance. It's all about balance. Okay. All right. So this is a good introduction to viruses. We'll talk about how HIV works on Tuesday, because HIV is not a DNA virus. HIV is an RNA virus. But we will talk about that on Tuesday. We do not have a guest speaker next Wednesday. Coincidentally, she's sick. So we will